Hello, everyone. I would like to use our time this evening to introduce you to the fascinating world of mathematical oncology. If you can't tell by the name, mathematical oncology is the study of cancer using mathematics. Typically, this involves making and using models that improve understanding of how tumors grow, how they develop, how they interact with the immune system, how they might respond to different kinds of treatments. And so this evening, I'm going to introduce you to one such model that I find to be particularly interesting because it originated in a very different branch of biology. It comes from the field of ecology, which studies how organisms interact with each other and with their environment. And yet, this model's relevance for helping us to understand how the immune system, like a kind of predator, is able to affect the prevalence of cancer cells within a patient's body. And so the goal of all of this is to ideally better understand the immune system so we can develop better treatments to help patients. Before we dive too deep into the context of cancer, I first just want to look at ecological theory and predator-prey modeling so we have the proper context. So predator-prey modeling basically begins with the Lotka-Volterra model, which was first developed in the 1920s. In its simplest form, it assumes unlimited resources, and it's given by this coupled system of two nonlinear ordinary differential equations. And so to talk through this real quick, we basically have the first equation that the rate of change of the prey population is equal to Rn, which is basically the current number of prey times some growth rate, minus ANP, which is the rate at which prey are removed from the population due to predation. And our predator population, its rate of change is equal to BANP, basically the amount of prey they kill multiplied by how efficient they are at converting the energy they get from those prey into predator offspring, minus NP, which is their natural death rate. So the Lotka-Volterra model predicts that both the predator and the prey populations will cycle between periods of high abundance and periods of low abundance based on their effects on each other. As you can see in this graph, which gives just a sample solution to a Lotka-Volterra model. And you can see how the population sizes oscillate. And I think this cycle makes a lot of sense just based on what we know about predator-prey dynamics. When you have one of the blue peaks, that means you have a lot of prey, which means there's a lot of food for the predators. So their population grows with the food. But then with more predators around, that of course begins to diminish the prey population because they all get eaten. And then with fewer prey around, the predators starve, which then allows, well, the predator population will decrease, which allows the prey population to grow once again. So that's kind of where this whole cycle comes from. But of course, in nature, species interactions are a lot more complicated than this simple model because there are so many other factors such as food availability, competition, disease, even the existence of other predators that influence population dynamics. And so over the past century, there's been a lot more models developed, more sophisticated models, to predict the effects of those other factors. And so that gives ecological predator-prey theory a very rich history that makes it very useful for applying to other contexts, such as cancer research. As I was researching this topic, one of the things that stood out to me the most was just realizing how much a microenvironment inside of someone's body behaves just like an ecosystem in nature. But of course, in nature, we have different animals interacting with each other and with resources like a river, a fruit tree, different kinds of minerals. But in these microenvironments inside of someone's body, we have different kinds of cells interacting with each other and with resources like glucose and amino acids. And so when we apply predator-prey theory to the microenvironment of the tumor environment, the predators are, of course, the cancer-killing immune cells, most commonly cytotoxic T cells. I'll just be referring to them as T cells. And our prey are, of course, the cancer cells that are being killed by the immune system. And so these two interactions, the predator-prey interactions and what I'll call the tumor-immune interactions, they do have a lot in common, but clearly they're still very different contexts, and that does lead to some key differences I want to highlight. First off, T-cell proliferation is not directly linked to cancer cell death. This is unlike predators in nature, whose growth rates are entirely dependent on how much prey they catch and consume, because that's how they get all their energy and their nutrients. But T-cells, they don't actually eat the cancer cells, they don't consume them, and so they don't get any nutrients from them. And that really causes a disconnect between the growth of the T-cell population and how many cancer cells they kill. But it is interesting that it turns out that there is an indirect connection between these two still, because increases in T cell population, they're primarily caused by external signaling pathways that promote the differentiation and proliferation of the relevant T cells. And these signaling pathways are more likely to be activated as more cancer cells are killed by the immune system because of the molecules it releases and everything like that. So there's an indirect connection, and I think, once again, I just want to point out how interesting it is that this behaves so much like an ecosystem where you have different types of cell populations influencing the growth rates and decline rates of the other cell populations. Secondly, I want to talk about competition. Because just like any ecosystem in nature, the resources within the tumor microenvironment are limited, and that means that T cells and cancer cells, they're not just in a predator-prey relationship, but they're also competitors. They compete for limited amounts of glucose and other nutrients like that. And so this extra element of competition is another way in which tumor-immune interactions deviate from the typical predator-prey interactions. So this difference definitely has significant ramifications for cancer research. And so I do want to show you one model here. It's still a very simple model compared to lots that are actually used, but I think it's helpful for understanding how the predator-prey theory can be translated from nature into the tumor microenvironment. So I don't have time to go through all the details of this, but I do just want you to see the overall similarities between this one and the Lotka-Volterra model we looked at first. That's on the right there. So the first two equations, the one for the cancer cells and the predator and the prey population, are actually pretty similar. I highlighted one term that's pretty different, and that one, 1 minus c over k, it basically limits the growth of cancer cells based on the availability of resources, which is more realistic. Our second two equations for the T cells and the predator population, these are fairly different. I'm just going to call your attention to the one I highlighted there. And that one basically accounts for the death of T cells, not because of the natural causes, but because of the cancer cells. And that's going to be the one there that accounts for competition between these two. So, having looked now at the major similarities and differences between predator-prey theory and tumor-immune interactions, the natural next step is to consider how these insights could be applied to cancer treatment. In ecological terms, the goal of treating cancer is to eliminate the prey population, or at least to limit its growth as much as possible. Now, when there's similar situations like this in the real world, where there's some species that humans don't want there, they tend to release a predator or a competitor into the ecosystem to try to eliminate it, or at least not let it take over. And so in cancer research, that's most similar to the approach of immunotherapy, which refers to a variety of strategies aimed at strengthening a patient's immune system to better combat cancer. And this makes a lot of sense, right? If we increase the predation rate of the immune system, we should be able to decrease the population of the prey, in this case, the cancer. Well, immunotherapy in a lot of ways still is not successful as anyone would like it to be, which I think leaves room for the new perspectives that ecological theory can offer. So the first thing to consider when trying to figure out how cancer could progress despite immunotherapy is the ecological concept of the Hydra effect. Hydra effects refers to the counterintuitive phenomenon in which an increase in the mortality rate of a population actually increases its size. When talking about tumor-immune interactions, though, what's more important is the evolutionary Hydra effect. That describes how increased mortality can accelerate the evolution of a population, which helps it better survive in its environment. An example of this would be if more individuals in a population die because they were eaten by a predator or something, that leaves more resources for the other members of the population to use. And then they can use those resources to produce more offspring more quickly. And then you have a shorter generation time that allows or makes adaptation more likely in a shorter time frame, thus accelerating evolution. But this process tends not just to be random, because predation also exerts a selective force on the prey population. It is typically the less biologically fit prey, the slower ones, the weaker ones that get eaten by the predators, while the more fit ones, the stronger ones, are the ones that escape and pass on their genes to the next generation. And so it is likewise possible that the immune system can actually enable tumor growth and select for cancer cells with adaptations that make them more likely to avoid immune defenses. And as a result, cancer progression typically reaches an escape phase in which the immune system is incapable of preventing tumor growth. Understanding these mechanisms, I think, is very important. Understanding these mechanisms that enable predators to actually enhance the likelihood of the prey population surviving I think is helpful for understanding the risks associated with immunotherapy. Now, on a more hopeful note, ecological theory also suggests how the actions of different types of immune cells could be maximized different types can be combined to maximize cancer elimination. Unlike different species of predators in nature that compete for their prey, the immune system actually has several types of specialized cells that actually work together in their efforts to fight cancer. And if these specialized cells differ in how well they fight cancer at different densities of cancer cells, an ecological concept known as their functional response, then it may be possible to combine these different types of immune cells into one immunotherapy strategy to better control cancer growth at a wider variety of tumor stages. Now, using ecological theory to understand the effects of immunotherapies certainly does not resolve all the remaining questions about how to improve cancer treatment, but I think it does provide a potential framework for better understanding how the immune system interacts with cancer, which could then help researchers to design more effective immunotherapies. And so, to conclude, predator-prey model is just one of the many different types of mathematical models being used to study cancer, but its well-laid theoretical foundations in ecology makes it a particularly useful one because so much of the theory is already there for researchers to just apply to what they need to apply it to. We've seen that despite the overall similarities between predator-prey and tumor-immune interactions, though, there are several significant differences between them that must be considered when using ecological insights to develop new mathematical models or immunotherapies. Thank you. It's also hard because you have to make sure your T cells also have the nutrients they need. So if you're starving out everything, you're also starving out your predator. So that'd be one thing to consider from this framework. Of course, a lot of models, if they're trying to account for everything, will include a lot. I've definitely seen models that are more competition-based, and there's definitely some things learned from the parasite relationship too. Yeah, I had to focus on one thing, but there's so many models you could choose to look at. And some people do wonder if it is more valid just to look at them as competitors and not so much as predator and prey. Yeah. I did read somewhere just about trying to keep accurate data and up-to-date data and taking, samples more often to get a sense of what is actually going on. But other than that, it would just be kind of more the theory of designing immunotherapy.